Hi. Today's lecture topic is going to be Chapter 4, A Tour of the Cell. I don't know what just happened there. Kind of lost my mind. So we're going to go through the different cell structures. It's just going to be sort of a definition kind of, there's not a lot of process here. It's just sort of what is this and what does it do? So it shouldn't be very long. It's not very difficult. It's just sort of a definition kind of thing. Where it is, what it does, etc. So switch to the PowerPoint and get started. So this week, we're going to review the structures of a cell, and we're going to do generalized cells. We're going to do a prokaryotic cell, a simple bacterial cell. We're also going to do a plant cell and an animal cell so that we can sort of see the differences in different structures that plant cells have that animal cells don't have, and vice versa. So how do these things come about? In 1665, a researcher named Hooke examined a piece of cork under a microscope and saw what he called little rooms. Prior to this, nobody knew that the larger organisms were made up of smaller cells, right? The cell theory didn't come until after this. Van Leeuwenhoek was working at the same time, and he created different lenses that could look at different types of living cells: blood, sperm, pond water. Now, of course, now we have many, many, many different types of microscopes: electron microscope, light microscopes, transmission microscopes, and most microscopes can see in the yellow area. Most microscopes can see between 1 and 100 micrometers. The eye can see almost down to 100 micrometers, right? And then below that, without an electron microscope or some super high-res microscopy, it's hard to see smaller bacteria and all the way down to individual proteins, etc. And basically, a microscope's job, right, is to magnify something so that you can see something that's too small to see with the unaided eye. Now, prior to 1665 and microscopes, right, we didn't have a cell theory. After the microscope was invented, we started to get a little bit more clarity on the fact that all organisms are made up of cells, and that's where we came up with the cell theory. So here we have a bunch of pictures on the left, which is light microscopy, and the pictures on the right, which are electron microscopy. Just different ways, different microscopes, highlighting different structures. You can see some of them are colored, some of them are fluoresced, some of them are see-through. On the right-hand side, you can look at the cilia and see the scanning, right, where you can see the outline of the outside, or transmission, we can see a cut right through the middle of them. So there's all kinds of different ways to look at microscopic specimen at this point. Different types of Nomarski contrasts, fluorescence, all kinds of things. If we were in the lab, we would basically just look at bright field stained, and phase contrast. That's pretty much all that we would be able to do with the light microscopes we have in the lab. Anything else is really expensive, really high tech that we just don't have in the lab. But we would have been looking at them with bright field, stained and unstained, and then a phase contrast. So when we talk about cells, right, the big question is always, why are cells so small? Why isn't a human being one giant cell? And really, it comes down to something called the surface area to volume ratio. The higher that number, the more efficient the cell is going to be in providing nutrients, removing waste, etc. So if you look at the big cell on the left, it's 3 by 3. Let's say that's 3 centimeters. 3 by 3 centimeters. That means its total volume is 27 centimeters. If we have the nucleus or something in the very middle of that cell, it's going to take a long time for the nutrients to go from the outside all the way to the middle. On the right-hand side, though, we have the same 27 volume units, the same size, but the surface area is now almost 3 to 4 times as large. So that means anything that needs to get into those cells has 3 to 4 times more surface area to penetrate, meaning the surface area to volume ratio is now up to 6. And so the higher the ratio, the easier it is for cells to exchange things with their environment. And the faster you can do that, right, the faster you can get things done. So cells being so small benefits the organism because each cell can get fed very quickly and the waste can be removed very quickly. If we were one big cell, the amount of time it would take for waste to go from our stomach area out to the environment would take forever, and it would almost be impossible to do. So size, the surface area to volume ratio counts and is really important as to why cells are so small. All right, when we think about cells, we want to start with the major differences or the major different types of cells. And since every living thing is made of cells, there are different structures that are common to all cells, no matter whether it's a bacteria or a grass or a tree or a shark or a chicken, right? And one of those things is the plasma membrane. So this is a picture of the plasma membrane. And remember from the last chapter on biomolecules that the plasma membrane is made up of phospholipids with a hydrophilic region, meaning water-loving, and a hydrophobic region, meaning water. So the plasma membrane is made up of phospholipids. You'll notice there are two phospholipids. There's the hydrophilic, the gray, and then the gray with the yellow sort of fuzzy fingers in the middle. Those are the fatty acids. And the plasma membrane also, as you notice, has proteins in them, some carbohydrate side chains, and we'll dive into the structure of the plasma membrane much more in the working cell, chapter 5. But you want to understand that the plasma membrane is a membrane surrounding the cell. It separates the cell from the environment. It allows certain molecules in, and it allows waste products and certain molecules out. All cells are surrounded by plasma membrane, whether you're a bacteria or any other type of cell, human cell, doesn't matter. This is one of the structures that makes the unity of all cells. The other thing that allows cells to be unified is something called the cytoplasm. And the cytoplasm in this picture, right, is going to be sort of that beige fluid inside of the cell. Here as well, it's that beige fluid inside the cell. And basically, it is the medium that all the other organelles float around in. So it's sort of like a semi-fluid. It's almost like egg white. So you'd have the plasma membrane with some egg white in the middle, and then that's the cytoplasm. So we have the plasma membrane, and we have the cytoplasm. That was me stopping for a phone call. So here we are. What were we talking about? We were talking about cytoplasm. So cytoplasm is the semi-fluid medium that's inside of every cell, supporting the internal cell structures. And then from there, all cells are going to have a variety of organelles and cell structures inside of them. Now, this picture that you're looking at here is a fictitious cartoon picture on the left of an E. coli bacteria, and on the right is an actual transmission electron microscopy of a actual E. coli. And the bacteria, because they exist as a single cell, they need some protective things to help them in their environment. So here we have the capsule on the outside, then they have a cell wall, then they have the plasma membrane. They have 3 layers of exterior coverings, unlike animal cells, which just have the plasma membrane, and plant cells that have a cell wall and plasma membrane. Bacteria have a capsule. Some of them will have fimbriae, which is those little finger-like extensions that help them to grip onto things. Some of them have flagella. They don't all have flagella. They don't all have fimbriae, right? But this is sort of giving you an idea of the prokaryotic cell structures. So prokaryotes, they lack a defined nucleus. The DNA is not inside a nucleus, but it is in what's called the nucleoid region, just sitting around inside the cytoplasm. What else? They are usually smaller than a eukaryotic cell. Usually they're unicellular. Most of the time they don't need oxygen. And again, no membranous organelles. What do they have? They have the plasma membrane, the cytoplasm, cell wall, the sticky capsule, nucleoid region of DNA, and ribosomes. Now, the only thing that's sort of new here is ribosomes. And ribosomes are the cell structures that create proteins. So any cell is going to need to have a process for creating proteins from the DNA recipe. So every cell is going to have a ribosome. Every cell is going to have a plasma membrane. Every cell is going to have cytoplasm. And every cell is going to have DNA. From there, we start to get a little bit more complicated, right? So we start to get a little more complicated. So which cellular structure is common, right? Nucleus? Nope. These other things you've never heard of? Nope. But the cell membrane? Yes. All three domains, bacteria, archaea, eukarya, all have a phospholipid membrane. So this is a picture of a fictitious animal cell, and you will see how it is much more complicated than the image of the bacterial cell. Eukaryotic cells are partitioned into various functional components. So each of those colored structures have a specific function. It's called compartmentalization. And there are membranous and non-membranous organelles. So things that are membranous are going to be the ER, the rough and smooth endoplasmic reticulum, the Golgi, the nucleus, the mitochondria, the chloroplasts, the lysosomes, the peroxisomes, are all going to be membrane bound. The other ones, such as ribosomes and different microtubules and microfilaments, are not membranous. So we're going to start with the structures inside of an animal cell. You can go to the Pearson and watch the BioFlix tour of an animal cell. It's a great way to sort of get an idea of what's going on inside of a cell as the cell moves around. We are also going to discuss structures that are part of a plant cell. This looks very similar: rough and smooth endoplasmic reticulum, the nucleus, the Golgi. The difference things here is, one, they have something called a central vacuole that stores water, and two, they have chloroplasts, which convert the sunlight energy into sugar energy. And we're going to go through all of these things. All right, so starting at the top, we have the nucleus. The nucleus is where the DNA is stored. The nucleus is surrounded by a nuclear envelope, a double membrane, and it allows for certain things to pass in and out of the nucleus. All right, and so the nucleus is where the DNA is stored. All eukaryotic cells have this. No bacteria and no archaea have this. So prokaryotes, no nucleus. Eukaryotes, a nucleus. From there, we have something called the rough endoplasmic reticulum. Now, the rough ER is the blue wavy stuff that's on the outside surrounding the nucleus. It makes sense that it's there because the point of the rough ER is to make protein. And you make protein, you need to take the recipe from the DNA and make the protein. So let's put that protein machinery right next to the nucleus. So it makes membrane. It also makes secretory proteins. Right, so here we have the rough ER on the bottom. The smooth ER is that tubular structure on the top left. Now the smooth ER is all about synthesizing lipids, fatty acids, phospholipids. So both the rough and smooth ER's job is manufacturing. Here we have the Golgi, this reddish orange looking folded membrane, the Golgi apparatus or the Golgi body. The job of the Golgi is to finish, sort, and ship cell products. So if you see number 1 there, you see a vesicle from the endoplasmic reticulum goes into the Golgi and then out the bottom side, right? The transport vesicle from the Golgi. Where is this going? So it makes a decision as to where these proteins and fats and lipids and things are going to end up inside the cell. Sometimes it's inside the cell, sometimes it's exported to the outside of the cell, but the Golgi is going to determine that. One of those exportive type vesicles is called a lysosome. And if we fill the lysosome with enzyme, it can participate in digestion. Right? So that's one of the sort of specified membranous organelles, but it comes from the Golgi. So let's see. Next. So if we're going to make membrane lipids, where are we going to do that? Right, membrane lipids are made in the smooth endoplasmic reticulum. That would be letter A. All right, so that's sort of manufacturing. Now we're going to get into energy processing. So there are 2 energy processing organelles in plant and animal cells. Mitochondria, these little kidney bean looking wavy things, is where we're going to take sugar and create ATP from it. All eukaryotic cells have a mitochondria, plant, animal, fungi, protist, right? It's going to be a double membrane structure, an inner membrane and outer membrane. But basically the role of the mitochondria is to convert sugar into energy. The chloroplast, only found in plants, has the opposite effect. It takes sunlight and creates sugar. So we have sunlight hits the chloroplast. The pigments, those green pigments, help to convert um, sunlight into sugar. Fascinating. So we're going to go over photosynthesis in chapter 7. So we're going to get to this, but this is a really, really interesting energy conversion where we're taking the sunlight and converting it into chemical energy of sugar. All right. Which one do plant cells not have, right? Plant cells have all of these. Plant cells have mitochondria, plant cells have nuclei, and plant cells have rough ER. Many times people believe that a plant cell has a chloroplast and an animal cell has a mitochondria. However, animal cells do have mitochondria, but plant cells have both. Plant cells create sugar from the sun, and they use that sugar to create ATP in their own mitochondria. So that's an important thing to distinguish. This right here is something called the endosymbiotic theory. And one of the big questions when you're studying cells is, how did cells come to be? How did the cell come to be so complicated on the inside? And one of the things that they kind of go with is something called the endosymbiotic theory. Endo meaning within, symbiotic meaning living together. And what they're thinking is that there was some sort of ancestor eukaryotic cell that engulfed a mitochondria, that engulfed a chloroplast, and created a cell that could now do multiple jobs. Endo within, symbiotic theory. All right, so this is a picture of all 3 types of cytoskeletal fibers. There are 3 different types, and there are 3 different roles. We have the microtubules, the microfilaments, and the intermediate filaments. They're all about structure, right? The microtubules are about maintenance of cell shape and some movement, moving the chromosomes, moving the organelles. Microfilaments, cell shape, muscle contraction, cell motility, cell division. Intermediate filaments, again, cell shape, anchoring things together. It's a little bit more structurally sound. So each one of them has sort of a different function of the cytoskeleton to sort of support the inside of the cell, because the cytoplasm itself is very liquidy, and if there was nothing inside, like an internal structure, the cell would collapse down upon itself. These are just pictures of the flagella and the cilia. That is a sort of a working interpretation of some of the cytoskeletal fibers. Here's an interesting question. So Taxol is a drug that's approved for treatment of breast cancer. It prevents depolymerization of microtubules. So if you're going to polymerize something, you're going to make a polymer of it, you're going to grow it. If you're going to depolymerize, right, it's going to break it down. And so the function that affects the cancer cells more than the normal cells might the Taxol interfere with. The Taxol's job, right, is to stop the microtubules from doing their job. So if it's going to interfere with the chromosome movements in cell division, that's going to mean that the cell cannot divide, right? So here we are, microtubules. What does a microtubule do, right? Cell motility and chromosome movement. If we're going to prevent chromosome movement, we're going to prevent the cells from dividing. If we prevent the cells from dividing, we are going to be able to prevent cancer from spreading, right? That's how Taxol works. And that's what one of the things we really want to start to understand when we're doing this study of biology. Why do we need to know this? Why do you need to know about microtubules? Well, because when you stop the microtubule function, you can stop cell division. Why would we want to stop cell division? We would want to stop cell division because we want to prevent cancer from dividing and spreading. So all of these things, right, even though they seem like little and complicated, right, it's important to know. All right, so the last couple of things we're going to talk about are the things that plant cells have that animal cells do not, right? We have a chloroplast, we have a central vacuole here. We have something called a plasmodesmata, because on the outside of a plant cell is a cell wall. That's how plants are allowed to stand up. That's why they snap when you bite into a carrot and snap a celery and break a stem, right? They are firm cellulose little compartments, and that's called the cell wall. Animal cells don't have that, right? We're very soft. Cell wall. Central vacuole is for plants to store water. Plants can't move and drink, right? So they need to store water when they can get it. They can't move. So central vacuole stores water, waste, pigment, etc. The cell wall allows for a rigid structure that the plant can build. And plasmodesmata are communication ports between the cell walls of different plant cells. All right, lastly, what we want to talk about is something called the extracellular matrix. When we talk about the extracellular matrix, we're talking about what's going on outside the cell. So here we have the plasma membrane, that gray and yellow wavy line in the middle. Inside the cell is orange, outside the cell is blue. You can see that outside the cell is very complicated. There's collagen and fibronectin and proteoglycans and all kinds of things. Now remember, in eukaryotes, most cells don't exist alone. We need to connect them together. So the extracellular matrix is going to do that. It connects cells to each other and holds them in a tissue. Right? So the extracellular matrix is also important to understand. Here we have different junctions between cells. Again, if we're going to hold them in a tissue, tight junctions, intermediate filaments of the desmosomes, as well as the gap junctions. Each one of them has a specific role. Each one of them holds certain types of cells together, and they perform different functions. So you'll want to make sure to kind of read up on that. According to the endosymbiotic theory, which of these organelles were prokaryotic that were engulfed, right? It's going to be the mitochondria and the chloroplasts. Why is that? Because mitochondria and chloroplasts have their own DNA. Interesting, right? They also divide in a different way, which we'll get to cell division in chapter 8. And they behave as prokaryotes within a larger cell. It's fascinating. So read up on that. All right, so the end of this chapter, it was a breeze through of the different topics of the cells. So make sure you really get in there and read the notes. Good luck on the homework. Good luck on the quiz.